Obrigado a todos por a presença aqui, presença online. E eu vou dar a minha aula em inglês. E a razão é porque toda a minha formação científica foi em inglês. Eu fiz a graduação, pós-graduação, doutorado, tudo em inglês. Então, é melhor para vocês se eu falar em inglês. E também na parte de perguntas, sem problema, pode perguntar em português e a gente navega as perguntas também. Se tiver alguma pergunta também aqui da audiência, durante a apresentação, não se preocupem, pode interromper. E a minha apresentação, eu vou tocar em várias áreas da cultura 3D. Então, eu vou cultura 3D a cultura 3D magnética como uma ferramenta para ajudar a fazer cultura 3D, mas também as áreas que estão mais avançadas agora, que vão ser desenvolvidas, como microfluidics e algumas outras aplicações. Tá bom? Então tá. I'm going to switch to English now. So we're going to, mostly what I'm going to talk is about results on magnetic 3D cell culture. And I always like to start with this quote here from Einstein, right? Everything should be made as simple as possible.
Uma nova patente para as áreas externas de seu lar, enquanto diminui o consumo de energia de sua residência.
Pessoal, uma das decisões mais importantes, elas estão... aliás, essa decisão, né, a decisão mais importante.
Por favor, se vocês estiverem online, não esqueçam de mute, tá bom? Para não dar o background, por favor. Okay, let's start over. All right, so I like this quote for a few different reasons. So yesterday we had a perfect background from Natalia, from Leda, and Suzani, why 3D cell culture, why it's relevant, why we're moving from 2D to 3D. And my experiences in human nature, whenever you have to add complexity to a process, you add the complexity, you have the new results, and you start to backtrack. You try to go back to make things simpler and simpler. And then you turn out right back where you started. So we want to make things simple. But if you're going to add complexity in the 3D world, 3D cell culture, there is a limit for you to simplify the process. And it's that simple, the way I see it. You make the organoids, the 3D culture tissue, and if you simplify, you're going to end up with the same problems with 2D cell cultures because your organoids are too small, you lose gradient, you lose cell-cell interaction. So you got to be mindful that you have to, complexity is part of the process. Okay. So the value of 2D cell culture, the way I see it, it's basically the 3D environment where cells can interact with each other and create an environment that is more predictive of in vivo biology. And as far as 3D cell culture, and for you that are new in the field, there is a lot of terminology, you know, and people call spheroid, organoid, organotypic, tumoroids, assembloids, there's all, the cells don't care. They don't care. All that matters is the biology. So cells are agnostic of terminology. So the key is predictive results. So, and that's where I think magnetic 3D cell culture really helps. It's a tool for you to manipulate cells in a way that you can, you know, you can manipulate, you have control of the variables to create a predictive results. Okay, so talking about predictive results, this and the QR code here is active. If you guys wanted to take a picture and get it, this is a issue from SLS Discovery on 3D cell culture approach for microphysiologically relevant models. And there's a very good collection of articles. There are 7 articles there. The editorial that also gives a very good view on where the field is and address a lot of tools that are enabling tools for 3D cell culture. And it's open source, so everybody should have access to journals. I'll let you take a picture quick. And the URL is right there on the bottom there, so right there. If you want to, yes. Okay, Suzanne just told me that she's going to put it on the site too, so you guys can just click and get the articles. All right, so a little bit of background where the technology came from. Of course, this is the foundation of magnetic 3D cell culture. Even though we're part of Griner, it was science. It was I was at MD Anderson Cancer Center, another collaborator of mine, a friend of mine at Rice University, and that's where we learned how to magnetize cells, use magnetic field to create 3D cell culture. So, and when you're talking about 3D cell culture, if you're doing science, a question that we always got from the beginning, and that's the value of 3D cell culture relative to other tools that we have for diagnostic, is function. Whenever you're publishing, you show your beautiful picture, the reviewers are going to ask, is there function? And I like this one because function for cardiomyocytes is very easy, right? So you can see that, you know, these are iPS-derived cardiomyocytes. You bring them together and they start beating. So, and these cardiomyocytes beat it for 20 days. So it's that's what we want. We want to take cells, bring them together, and create an environment that that is reproduced biology in vivo. All right. So next turn here. So as you guys heard yesterday, Natalia gave a great review on the different tools and techniques. But from a practical point of view, when you go from 2D cell culture to 3D cell culture, you actually appreciate how valuable it is, cells attached to plastic. Because cells are attached to plastic, it's easy to change media. Simple, you just take the media out. You want to do the downstream work. Immunofluorescence, they all attach to plastic, you don't lose your cells. So when you go to 3D cell culture, that's lost. And it's no longer easy. Like the work that Leda showed yesterday, changing media in heavy long-term culture, it's not trivial when you're doing 3D because the cells are not attached. So having a way to manipulate the cells is very important. And what we do, and that's how we think of magnetic 3D cell culture. The magnetic field is a surrogate to cell attachment. So we can put the magnetic field, hold things down, or bring cells together and remove, and it's back to normal. So, and of course, as Natalia described yesterday, and we magnetize cells. So we have an nanoparticle that is just electrostatic attached to the cells, and use the same workflow as you do with normal culture, but you can put the magnetic field on top and levitate the cells off the bottom, or you can put the magnetic field on the bottom and shape, and depending on the shape of the magnetic field, you can actually create shapes. And I'll show you some data that it's actually, you know, the term bioprinting is accurate because we can print things. So maybe I'll turn it down a little bit, light. No, wrong way. So this is just a quick examples of some of our results. Perfect. So, and because we magnetize cells, apply the magnetic field, the cells come together fast. So here you have, you know, just a spheroid that you assembled in 12 hours, and you're probably going to see this today on our hands-on results earlier today. Here we published this in Nature Nanotech. We actually took the GFP labeled glioblastoma with normal human astrocytes, MTR labeled. We cultured them separately, and then we magnetically brought them together. And you can actually see the green, which is the glioblastoma, just takes over the culture. It just migrates, and which is, you know, if you're thinking metastasis, that's probably the mechanism of action. If you add a drug and you can interrupt that, that's a good diagnostic. And I particularly like this one too. This is an adiposphere. This was actually the cover of Tissue Engineer a few years ago. And I think it's very relevant because especially from talking to Flavio and Suzanne, there is a lot of emphasis in this department as far as regenerative medicine, right? So, and one thing that I forgot to say, I'm a chemist. So I think of cells in simple ways. They're smart. You know, you bring them together in the right conditions, they do what they're supposed to do. And this is a good example because when we started doing a magnetic 3D cell culture, we have this collaborator that wanted to create an adiposphere, but they wanted to also include the angiogenic process of the tissue. So they want to incorporate it into endothelial cells. So they knew how to differentiate the pre-adipocyte stem cells, and they came to me and was like, okay, what ratio of cells do we use to create an adiposphere? We want to see the angiogenesis, the cell sprouting. And easy answers for me was 50-50, you know. So we do 50% pre-adipocyte stem cells, 50% endothelial cells, brought them together, they integrated, but they did not form the angiogenesis process. So we're like, okay, what do we do? So we played with the ratio. You have to optimize. When we had 5% endothelial cells, 95% the pre-adipocyte, we got the green here is basically the sprouting, is GFP available endothelial cells. The red is pure lipin. So when we played the ratio, they just did what they're supposed to do. And we had this beautiful culture. Okay, so from that, we had another hypothesis, right? We're starting. So again, thinking of cells being smart. What about if you take the exact ratio that is in vivo? What do the cells do? So the approach we took the mouse. These are fat mouse. We removed the adipose tissue and we took the whole entire stromal vascular fraction, all the cells. So in principle, that's how it is in vivo. That's the right ratios, right? You magnetize the cells and we assembled the tissue. And this is what we got. So the green here is CD31, which is blood vessel marker. And the green on this side is decorin, which is stroma. The cells basically assembled into what it looks like blood vessels. And the decorin, which is the stroma, was right supporting the cast, this is what an organoid is, right? It takes a stem cell and or a tissue that has stem cells, that has stamina, and you create an organ-like structure. So again, if you bring cells under the right conditions, they do what they're supposed to do generally. So, and of course, there's media. If you're using different cell types, you have to take in consideration a lot of factors, but it can be done. So okay, so that's a good example of organoids, and that's a term that I'll touch in a little bit. And whenever we're talking about organoids, there is matrigel, right? Everybody that is doing organoid work at some point use matrigel or use matrigel, and there is almost a correlation that if you're doing organoid, you have to use matrigel, and that's not true. I just showed before we did the adipocyte, the organoid of the adipose tissue without matrigel, and this is a work that was done by a collaborator of mine at the University of Freiburg, and they actually compared the magnetic 3D cell culture matrizome, the amount of extracellular matrix proteins from the magnetic 3D cell culture versus the ones cultured in matrigel. And they use a beautiful work we published at Matrix Biology, which is a great journal, and they use mass spectrometry to determine to characterize the tissue. And one thing that was obvious right off the bat, the cultures promoted by cell-cell interaction with magnetic 3D cell culture, the number of peptides were much larger than matrigel. And more quantitative, this is the Venn diagram. So of the proteins that we found by mass spectrometry of the 3D cultures in matrigel and non-matrigel, the abundance on the magnetic 3D cell culture was much harder. We had 124 proteins that were not in matrigel. We had 27 overlap. You only see 6 that were unique for matrigel. And of course, they didn't stop there. Okay, you have cell-cell interaction, it's different, it's richer. But they have a very nice comparison of the relevance as far as the extracellular matrix from in vivo to the in vitro tissue, and it resembles much more the complexity that you have in vivo than matrigel. And they also look at their mRNA level, and you can see the log fold of the 3D proteins in 3D versus 2D is much larger as well in promoting when cell-cell interaction promoted. So cell-cell interaction is key. Matrigel is not always needed, and it's not always useful either. So all depends. The tool we're going to use, as Natalia mentioned yesterday, and as Leda showed yesterday, depends on the question you're asking, and then you choose your tool to find the predictive results. All right, so I'd like to touch on this because I'm sure maybe you guys are being polite, not asking, but we always get this question: what do nanoparticles do to the cells, right? So we have, for years, we have published a lot in very good journals, and they always ask, what do nanoparticles do to the cells? And we've shown over and over, it's bioinert at the concentrations we're using. Cells are very buoyant. So you just need a few nanoparticles to move them. I like to joke, you have a headache, you take an aspirin, your headache goes away. You have a headache, you take 30 aspirins, you may die. So it's very small amount. Concentration is what matters. So it's very small amount of nanoparticles, and it's basically bioinert. And we have, compiled all these results, viability, proliferation with, without nanoparticles, protein expression, stem cell differentiation, oxidative stress, inflammatory stress, genomics. So it's pretty much, it doesn't do anything. And also, and I'm going to show some results collaboration we have with Scripps Research Institute in Florida, where we just finished, just published 150,000 screening for pancreatic cancer cells. I'll show some of the results. And before we started, just like all the other biological aspects of 3D cell culture, the question was, do the nanoparticles interfere with drug interaction with cells? And we've done spin-offs of cell lines, different compounds, exactly the same dose response. It does not affect. So, okay, so the tool is very simple. You have your magnetizer cells, you put a magnet, they come together. You do not need to leave it on the magnet, and the cultures assemble. I'm not going to show the movie that Natalia showed yesterday, but I'm going to show a different movie. We have a new tool that we came up with, which is up to facilitate moving things around. I think they would be very helpful for the work we did, which is the multi-mag pen. So again, as I said, changing media, manipulating cells, it's not trivial. So we came up with a tool that's basically a magnet on a sleeve that is treated and it's sterile, and also we apply a cell repellent treatment so the cells don't stick to it. But you can very easily now take 96 spheroids and transfer from one plate to the other. Very easily. And here's the movie. It's a cartoon, but it's a quick movie just for us to go through. So So you have your 96 well. You have two formats. You can have in 24 well or 96 well. And then you have a spheroid into the well, magnetized. It looks like a PCR plate, but it's treated. It's not that, not any PCR plate will work. It has to be self-repellent. So then you put your sleeve in, you put your magnet drive. This little shake is important. And then you can just transfer from one plate to a fresh plate. If you already have drugs, you can just put it right into the drug. You know, you can prepare, you don't have to. So, and then you take your magnet out. You put a magnet on the bottom to pull them off of the sleeve. And that's it. So this is a tool we just launched, and it works really well. We're thinking about going with a 384, but we're not quite there yet. So again, by magnetizing the cells, you can use this like a pipette, but it's very gentle. You don't disrupt the structures, and you can move things. Yes. Mm-hmm. Uh-huh. Boa pergunta. Então, a Suzane está perguntando, when we make the 3D culture, the cells replicate, and of course, the daughter cells leave the nanoparticles behind, because nanoparticles behind, so the ratio of cells to nanoparticles is always increasing. But most cells, they stay connected via extracellular matrix. So we actually, in the Nature Nanotech, we show that the nanoparticles get stuck in the middle of the extracellular matrix. So, and the cells just grow. And we have a patent process where we can create magnetized DCM, basically from any culture, by decellularizing the process. But some cells, though, they fall off. Then you may have to remagnetize. The only time that we have remagnetized the cells is with patient samples. We are trying to expand them because we're breaking them. And so we remagnetize them to keep expanding them. But generally, you don't have to remagnetize because they're stuck together. All right, so that's again, just showing the real data. I like the movies, the cartoons, but you know, you can really transfer everything very nicely and it works pretty well. So another tool that we have, sometimes you want to make multiple spheroids in the same well, right? So we can mix and match different magnets. In this case, we used a 6 well with a 384 magnet array. So we put the cells on the plates, and what happens, and this is real cells, a picture, you can create, you know, several spheroids in the same well. And again, if you play with the different size, different cell number, you can vary the size and control. So it's just, again, a tool for you to manipulate and make different structures in the same well. So now I'm going to go into work we did with a colleague of mine from MD Anderson, Jeff Bartolomeos. In this, we used triple negative inflammatory breast cancer. So here, they actually took the patient tumor and we grew the PDX. Then we took the PDX and we want to see how the question was, can we replicate the morphology, the marker distribution from the PDX into the magnetic 3D cell culture? So in this, what we did, so we took the tumor from the PDX, cut in half. Half we processed the way we do, the other half we came up with a protocol. We broke the tissue apart and we magnetized the cells and we reassembled. And that's the result. We look at Icadirin, Vimentin, Ki67, Pismat, but this is a good, it's not. So you can see this is the mouse, the PDX. It's a paraffin embedded section. and we did immunohistochemistry for Icadiri environment. It's not perfect here, but it's just because of the resolution of the monitor. But the middle and the right are the bioprinted. But you can tell the distribution of markers is very similar. And we did the gene expression comparison. Its correlation was above 95%. So we actually took the tumor, we broke it apart, reassembled. Now, the beauty here, now you can actually, we know it's so expensive to do with PDX. Now you can actually break it apart and test several drugs in a much higher throughput way. So this was great work that did. This was actually was another PDX we did with another collaborator, prostate cancer. In this one, we had we did a pretty big screen, and again, we just printed in a 384 well format, and it worked really well. And we're moving towards personalized medicine. This is actually a needle biopsy from metastatic site of prostate cancer, metastatic site, very small sample. We took that, we broke it apart, we magnetized, and we test doxorubicin. And we got a very nice dose dependence curve from reconstructing the tumor. Okay. Now, this is going through high throughput screening. This is the work we've done with Scripps. One of the papers, actually, the 150,000 compound screening is published on the SLS Discovery on the QR code that you guys got. And here we got a big grant from NIH, from NCI, to actually validate our 1,536 platform. So this is really dense. So yes, you need automation. 384, you can do in your lab. 1536, you need automation. So, but that's the grant we got. It was us, Cold Spring Harbor, the group of David Tuvzo, who is the president for AACR. And this is just showing that, you magnetize your cells, you put it on the well, the magnetic field brings all the cells together very quickly and assemble them. That's just showing the reproducibility of 1536. It's hard to see, but every well has a single spheroid, very uniform in size. And again, the size of the spheroid is defined by the number of cells. So, and again, enabling to 2,500s. 1,200 to 2,500 is the optimum Z prime that we get for, yeah. And so practical aspect, organoids imaging, you wanted to image, right? There are a lot of fancy tools for get beautiful images, great. But when you're doing imaging, you want to do on a flat surface. Not all 3D cell cultures that Natalia showed are flat. So if you want a high resolution, flat is physics. You need flat, okay? So the problem with flat, with the spheroids, there's thermal motion and the spheroids move. And as you can see here, this is a 384 well picture. And I'm trying to get the mouse here. So see, there are wells that there are no cells. It's because they move to the side. And whenever they move to the side, it's very hard to microscope to catch the image. So because the cell is magnetized, you can put on the magnet for 30 seconds and you recover all your spheroids. And they're also in the right place, which is very important. If you're thinking about high throughput, remember, the microscope is gonna, if you have 1,536, if it takes 30 seconds to take every image, you can do the math, how much you're gonna take for it to take the full picture of the plate. And if you have to look for the spheroid into the well, by the time you get the first picture, is a completely different time point on the last. So positioning is very important when you're talking about high throughput imaging. So it seems simple, but it's not trivial. This is something we're addressing, a real need. So, okay, now 3D cell culture morphology. And I'm going to touch again on the, organoid, spheroid aspect. So what you see here, there are some cell lines and some patient cells. So this work that we did with Scripps in Cold Spring Harbor it was to validate high throughput screening using patient-derived pancreatic cancer cells. Okay, so these were patient cells isolated by Cold Spring Harbor, and we took the pancreatic cancer tumor and we separated the cancer cells from the CAFs. The CAFs is the cancer-associated fibroblast, right? So they always live together. So just as a comparison for the different type of morphology you can get as far as 3D cell culture, you have HT29. This is ovarian cancer cell line. See, we magnetize the cells, bring them to the middle, and they form a beautiful spheroid. You take the PANC1, which is pancreatic cancer cell line, same thing, put them together, they make a very nice structure. Now, when you go to the patient samples, AGM1, H21, AGM1 CAF, and H21 CAF, AGM1 is the cancer cell of one patient. AGM1 is the nomenclature for one patient. H21 is another patient. And the CAFs are the associated one. If you look at the CAFs, which is you see today, the fibroblast, you bring them together, they make a beautiful spheroid easily. Okay, you'll see on the experiment, hands-on experiment with the fibroblast. But if you look at the cancer cell line, they don't form a cohesive structure. They make organoids. Organoids, again, usually organoids are organotypic-like structures that usually there's some stamenas that they created this particular structure that usually has a lumen, it has a cyst type of morphology, and that's what it does. There's no, usually, as I said, matrigel to create organoids, there's no matrigel here. The cells just make organoids, which is fine. We still can magnetize and you can assemble them and do all the work we do, all the benefits of magnetic 3D cell culture. Okay, so the outcome of this study. So I'm adding layers to the hypothesis of this grant that was funded. So there was the validation of the high throughput. Then we also, one of the aims, one of the hypotheses we had for this grant is, the hypothesis was that 3D cell culture is better predictive of drug interaction with the pancreatic cancer cells. So that was the hypothesis. So how do you prove that hypothesis, right? You take a library of drugs, right? So in some ways, you want to make sure that you have less false positives on the screen. So we took the FDA library, which there were no cancer prescribed drugs. So in a way, those should, whatever is positive should be a false positive, right? So, and that's what we did. And we compared 2D versus 3D. So when you look at the patient samples, H21, 3D, less hits. Those are basically the number of a 3,300 compound library. 42 were active in 2D, 26 in 3D, less. So all the 3D were less than the 2D. So in a way, again, the 3D we're getting, and again, if you're talking about thousands of molecules, if you can cut in half, that's meaningful when you go to the drug development process. It's meaningful. So now we dissected the drugs a little bit more, and which was nice as well. We split these results in this sort of 4 quadrants. So you have inhibition of 2D in the x-axis, you have inhibition in 3D in the y-axis, okay? So if every dot's a compound, if the compound has a sensitivity in the pink area, it means you only kill the cells in 2D. If it was on the white, it didn't do anything to cells, which is most of the cells were not active, which was expected. Then the green is the overlap 3D and 2D, but that we really appreciated was we picked one drug that was very active only in 3D, and it was disulfiram, which is it's acetaldehyde dehydrogenase, and it was in phase 1 clinical trial for pancreatic cancer too. So in a way, we actually picked the one that was actually working in vivo. So I was blind, no, we didn't know. I was just glad it was an aspirin. Yeah. So again, and it's a pretty nasty drug. It's a drug for I think alcoholism and drug addiction, it's not a very nice drug. So this was pretty cool. And then we just finished 150,000 compound screening, which to our knowledge, it was the largest one done, first time in with the patient-derived pancreatic cancer samples. We got about, 0.5% of hits. So this is published. There was a really nice work we completed. So, all right, on this work, a little bit of biology, a little bit of touch on mechanism, and everybody talks about 3D cell culture, cell-cell interaction, and extracellular matrix. The way I like to think, what comes first, DCM or the cell contact? Is the cell contact, then you have DCM. So, and that's why we always pay attention to cadherins. Cadherins are very important. So, on this work with the scripts, when we're looking at the organoids, we also trying to probe the mechanism. And we make the organoids, we have the fibroblast structure, very nice spheroids. So we wanted to sort of poke on the mechanism. And what we did, we decided to block the integrins and the cadherins with antibodies, just to see what happens, okay? And this is what happens. So this is the CAF, the patient cancer-associated fibroblast. We magnetize, we brought them together, and you make beautiful organoids. The control, that's fine. Then you take the cancer cells, they make beautiful organoids. Fine. Now you use anti-integrin. You're blocking the integrins. So you're blocking the extracellular matrix interaction. Look what happens. Not much. Doesn't do much. And this is a CAF, cancer-associated fibroblast. But if you, even with the, and again, same thing with the cancer cells, not much. But when you block the cadherin, they don't like to form, which makes, it's cell contact. Even with the fibroblast, it really interferes with the assembly. And it's the same thing with the cancer cells. You put the anti-cadherin, and again, cadherin is a lethal gene. It's very important. So the contact is very important to create the assembly process and then the ECM. So this was pretty cool. And again, that's why I said cell contact is key, promoting cell. And even, actually, I didn't mention that, but on the PDX work we did with MD Anderson, they wanted to they magnetize the cells and just put it on the magnet, right? For a few hours, actually, no, we did 30 minutes, took it out, the cells would not form. They had to keep it on the magnet and push them together to actually form the reproducibly, the PDX models or the printed models, because you're pushing the cells together. Gravity is not enough. All right, so another interesting, cool result, and this was also published on the SLS Discovery. As I said, we took the pancreatic tumor, cancer tumor, from the patient. We separated the CAFs from the cancer cells. And the question was, what about if we bring them back together? What happens? So, and that's what we did. We labeled the CAFs red, the cancer cells green, and we mixed them together. So this is the CAFs. The middle one is just the cancer cells. And then you have the co-culture of the two cell types. So remember, the cancer cells make organoids. They don't come together. But if you co-culture them, first thing they do, they all come together. They all interact, they all come in a single structure, and then the cancer cell comes out. So there's a lot of, metastases. There's a lot of cell migration. There's a lot of things, but it's very interesting. And again, this is what we detect. And that's another important part on cell biology, cancer biology. But we forget time. Time is very important. So if you do a measurement here, you're going to get a different result than you get it here. So longitudinal studies are very important, and we forget about that. All right, so let's see. Okay, getting some water. I'm going to take a little turn here, which is something that, again, motivated by the longitudinal aspect. Time is important. Managing the mask is not easy either, but it's necessary. Great. Sorry, broke my mask. All right, so now time in biology. So one thing that it's interesting with 3D cell culture, especially magnetic 3D cell culture, is that we have the ability of magnetizing cells and bring them together. And once you bring them together, if you start watching them, there's a lot of motion. This is very dynamic. The cells are moving. And again, it's not a round bottom, so it is cell-cell interaction. So the question we had when you saw this process is, can we use the cell mobility as a readout of biology? If it's drug interaction, if it's mechanism with drug interaction, if it's just a viability assay. And so that's what we did. We print them and you observe how the cells move. So this is it's a movie. We publish it in scientific reports, and it shows you how much the cells move when you bring them together. So here is 3T3 fibroblast, and we use retinoic acid, which is vitamin A, anti-wrinkle cream, right? Just as a toxicant. And every dot you see here it's freshly printed cells. You just brought the cells together, okay? So we have high concentration on the top, you have a control on the bottom, and look at the time here. This is the time of the measurement. So, 2 hours, 3 hours, see the control that got much smaller than the other culture. Oh, sorry. There you go. So if you look at 3 hours, if you look at the size of this dot on the top, it's bigger than the bottom. And the bars here are actually changing size that we're monitoring. So in 3 hours, you get a dose dependence curve, okay? But what is really valuable about this measurement, and I'll show you why, is the time-dependent curves. As I said, time is key. So we have time and the changing size as a function of concentration. Those curves, that's important. Every cell type has a different curve. And if you add a different drug, it has a different curve. So, and in this case, it's just death. The cells are dying, they're not moving as fast. And we show that there's just viability, like live dead. But if you take a pen of drugs, all the same cell type, the same plate, different drugs. And this is just a live dead. And if you look at the curves as a collection, so you have time and changing size. So this like high concentration of doxorubicin, cells are moving, they're dying. First clean, all different shapes. So you're almost looking at this motion and inferring to the drug interaction. So, and here is SDS, which is just dissolving. So the question is, can we use this to fingerprint to understand mechanism of interaction? And that's what we did a different way now. Now we have the same drug, different cell types. You have A549, which is lung cancer, HCT116 colon cancer, and HepG2. Look at the curves. In this doxorubicin, same plate, very different. You can see that there's a dose dependence. So what we just did a polynomial fit on this, and we use a PCA, principal component analysis, and we can actually separate the cancer cells, fingerprint this cancer cell, the type of cells, and the dose interaction by just looking at this motion of the cells. And we also did this with pancreatic cancer cells, which we mapped the cancer cells separate from the normal cells based on this motion. Now, for you to get this data, we took full 384 plate every 15 seconds. So an instrument to do that, it's not cheap. But the only reason it works is because, oh no, I'm going the wrong way. We use a mobile device. We use an iPhone. Because the nanoparticle gives the contrast. And we wrote an app that it can take the picture. And so it's not microscopy, but it's very sensitive. And that's the only way we could do it. And we published this, we have several papers on this, and it works. But you can do, if you have a fancy microscope, a scanner, you can use it. All right, so same idea, printing, I'm almost there, almost finished. We have, this is the bioprinting idea, right? And this is an assay we developed as a surrogate for aortic ring assay. If you're measuring vasoactivity, what people do, they take the aorta, they chop up the aorta, make the little rings and stretch, right? And it's not high throughput, it's not very reproducible, and use animals. So our hypothesis was with AstraZeneca, we did this in collaboration with them. Can we take vascular smooth muscle, print a ring, and see if we can watch the cells contracting for vasoactivity or not? And that's what we did. So we have a magnet that is a ring, and this is the movie. So this is a vascular smooth muscle, and every ring here is a ring of vascular smooth muscle. We use blebbistatin. Blebbistatin blocks phosphorylation myosin light chain and as a vasodilator. So if you have high concentration, remember they contract. The rings are going to close slower because of concentration, the high concentration of blebbistatin. So you have high concentration, you're going to have high concentration here, control on the bottom, and you're going to see the bars on the side, which is quantifying the structure. This is overnight. So watch. See, the control is closing much faster, and the blebbistatin is acting. So we can, quantify this process, which is, again, it's a needed tool, and this is the quantification of the process, which is, again, the movie is straight from the iPod. So, and of course, the biology, right? I said that it blocks phosphorylation myosin light chain. So you have the myosin light chain here, the immunofluorescence, you add blebbistatin, you block, and you see correlates to it. And we published this in scientific reports. We compared human and animal with a panel of drugs, vasoconstrictor, vasodilators. I say magnetic bioprinting. See, we can print. We can just use magnets and assemble the cells in different ways. So now I want to touch on something too, because microfluidics is the new paradigm, and we have a little bit more. Yes. It really depends on how many cells you want. Depends on the cell size, but in principle, you can just increase the number and have a thick layer. It's really the number of, because we're not relying on replicating the cells. So the number of cells you come in is what you have. And we've done this ring assay with adding endothelial cells, and it makes a difference too. So for the students and for us too, I'm going to touch on the microfluidics now. That's the new paradigm, I think, coming from 3D cell culture. Microfluidics is, again, as the name says, fluid flowing through microchannels. But now we want to add the 3D cell culture and see how that can facilitate and enable us to get better results. And this is in collaboration with a company called Protein Fluidics. We've done a lot of work. They adapted the magnetic 3D cell culture to their chips for exactly the same reason as we do magnetic 3D cell culture, to make it easier to enable things that we cannot do without it. So the system is pretty straightforward. They magnetize the cells, and they have these microchannels. They have a little magnet on the bottom that it can hold the cultures down. And basically, you really have these microchannels that it allows you to exchange fluid. And if it's to add, remove drugs, if it's to add, remove metabolites to extract to analysis, if it's to do immunofluorescence staining, those dependents, and I'll show you exactly how it does. This is a nifty movie. So basically, instead of using a 96 well, you have a microchip. So you put a spheroid in the chip. And now that's your spheroid there. And now you can control the fluid coming in and out if it's to replenish without touching it. And actually, I want the next movie. And that's how the process really works as far as the media exchange. So you have different wells, different, you can add your different, if it's a drug, if it's primary antibodies, fluorescence staining, you can control everything with the microprocessor inside the incubator, and you can exchange all the fluids and without really touching it. So it's a pretty neat concept. And now we're trying to work with them so we can make the cell repellent chip, so we can actually use the magnetic straight on and create these structures in there. And this is, again, this is a Hep2, just the toxicity assay that they did using statosporine and just a dose dependent, where they did the whole assay inside of the flow chip. So they can control the dose, and you can see the live dead just using toxicity of liver cells. This is our patient-derived tumoroids. Same thing, they did all the staining using the microchip. And this work actually was a collaborator from ours here. I think they went from Brazil to Boston, Rafael they also used the microchip to look at adipose tissue. And this is was very interesting too, because this is another enabling aspect of magnetizing the cells. And I knew that from our previous work using adipospheres. When you generate an adiposphere, fat floats. You cannot keep it in the bottom. How do you take the image? So they actually had to use the magnet to bring them down, hold them down, and then they could get these beautiful images. So here is a 2D morphology. This is the adipose tissue. And these images are really, really nice that they did. So again, it's an enabling tool. They couldn't get the image, by magnetizing the cells, they were able to do it. So I'm almost there. This actually, from our conversation yesterday, Flavio, I added this slide too. This work we did with a colleague of mine. He's actually in Thailand, João Ferreira. He's a dentist trained at NIH, and now he's in Thailand. His wife is from Thailand, but he has done some great work. And this work was pretty neat because they created salivary gland organoids from neurocrest stem cells using magnetic 3D cell culture. But they also reimplanted in the mouse. So he's a dentist. When you have head and neck cancer, right? You get radiation, you kill your salivary gland. You get dry mouth. So the idea here, can we take the stem cells and recreate the salivary gland and put in the mouse and create function? So they actually irradiated the mouse, the control, and reimplanted. And against the control, here you have irradiated mouse in the non-radiated mouse. And the black bars here are the epithelial growth index for the two systems. Every time and this is collected from the in vivo sample after they implanted. So they took the salivary gland, we implanted the irradiated mouse, and the tissue integrated and showed function. So that was pretty neat. And actually, they actually showed this is the organoid of the non-radiated. The non-radiated integrates beautifully. You can see reinnervation of the tissue, and but the radiated one also showed a lot of function. So that was pretty neat. And discussing too, we had done, this is a topic that it's a joke, but nobody cares if the no particles do anything here. So this was actually this was a serendipitous project, because just like I came here yesterday telling what I did. I was at the urology department at University of Texas Medical School, and we're just discussing some ideas. And one of the postdocs was describing from the group that I was talking to that they have the stem cells that he believes addresses nerve regeneration of erectile dysfunction. But they could not keep the stem cells. They had the stem cells that they said addressed, but they could not keep the stem cell inside of the rat's penis because of the dynamics. They cannot stay. And I asked, can you glue a magnet to the rat's penis? And they laughed, but I was serious. I was like, can you? And they did. They did the whole project, and the tissue integrated, showed function. They followed the cells. So again, it's enabling tool. Magnetism is great. And of course, I'm very proud of this. This is a work we did in the space station. This is Peg Whitson after she came back. This is her handling the magnets and the magnetized cells in space. And there are a few reasons why we did this project too. So again, the enabling tool aspect. If there is no gravity, cells grow in 3D. Why do we need a 3D cell culture in space? It's very hard to manipulate cells. So we send magnetized cells. So in astronaut time, it's very expensive. So if you can help them to save time when they're culturing cells, it's useful. So that's one aspect. The other aspect, nobody had created a monolayer culture in space. They have tried to fight the plates, but they don't make it because of vibration, the cells come off. So serendipitously, we wanted to, and we did. We printed the cells on the membrane. They stick, but they did not spread. They grow. It's interesting again, but it was fun, and we have some other ideas to do. And this was pretty neat work. And of course, we have lots of collaborators, and I'll be ready to answer any questions. And sorry, it was a little dense, usually more than I usually give, but I wanted to, take the time. We're all here, so to share as much as possible. And I can answer in Portuguese. All good questions. So who is it? Martin, good question. So 4 different cell types. Actually, we've done a few years ago was a lung tissue also. We did 4 primary cell types: epithelial, fibroblast, endothelial, and epithelial cells. You can definitely build layered or just mixing them together. As far as how big they can be, again, it goes back to what is the question you're asking. Yes, if you have a large tissue to culture over a long time, you're going to get necrosis. You're going to get nutrient impairment into the middle of the culture. So that's an issue. But that's how you have to plan accordingly. Some people want to have the hypoxic environment in the middle, especially if you want to replicate a hypoxic environment. But you can control that by the number of cells and time, usually.
First, congratulations. It was wonderful to see you talking about that. Congratulations. And I'd like to know about some influences of physics aspects of some of the physics aspects, like it's because some researchers come into the laboratory wanting to test this influence of physical aspects of, for example, electricity in the culture or light in the culture. Do you think it could help in these organoids?
It depends on the cell type, I think. Of course, from a chemist, or, actually I'm a physical chemist, so there's always the physical and the biology always being introduced. In the case of magnetism, there is actually a work from that just came out, I haven't read it yet, where, there is some claims the magnetic field can interfere with cells. But there's also difference between alternating field and static field, right? We use static, and often we don't leave the magnetic field on the cells. We put them together and we take them out. Now, if you're talking about mechanosensitive stem cells or mechanosensitive cells, if you're going to squeeze them, they're going to respond. Even with the cardiomyocytes. Sometimes we're coaching the cardiomyocytes, they're beating, they stop beating, we put back on the magnet, they start beating again, which is kind of makes sense. You're squeezing them together, you're getting those, ion channels, maybe to reconnect, and they start beating again. So, electric field, again, it depends on the scale, it will depend on the frequency, because if you think current, it's ionic current, right? Those are ions flowing. So it's not quite exactly as electric flowing. So, but in the beginning of cell phones, right, we thought that, radiation would create brain tumors and all that. I don't think we have enough cell phones that's probably not the case. So, but it would depend on the type of tissue and the type of cell, I think.
It's the same for the lights, like
light as well. There are a lot of people that use photodynamic therapy to study different things. And but it will depend on the type of cells. If the cells have pigments, they're probably going to respond more. Melanoma, they're black, they will respond to light faster. So I think it depends a lot on the physics of the cell itself.
I have another question. It's about mitochondria. The role of mitochondria in the cell is very important. And I'd like to know if you guys have chance to assess the mitochondria in the growing of the cells and if we influence, because we know that cell with a low amount of mitochondria has a potential to not grow. So
and that's a very active area. And later showed also, mentioned mitochondrial activity to see different pathways of, viability or solar path. From a practical point of view, this, like the microfluidic system, they're really going towards metabolomic analysis. So they can extract the supernatant, measure the amount of metabolic to monitor this. It's a good way to monitor the cells real time. But mitochondria is, and there's, again, I guess Agilent is the company that has the first one, I think, that addressed the Seahorse tool, which is for mitochondria, which actually we have a paper with them. That was also very hard to place the organoids onto their Seahorse system. And we use magnetic to pull them down and hold them there. But yeah, mitochondria, all types of readouts, reporters, that's a hot area too. If you can have cells and modify them, have some key reporters there, then you can have real-time longitudinal readouts. There's a lot of value on that. Thank you very much.